Hi, Lauren. I'm just going to give you an overview of this to see what you think. I think you could do this very well, and it might be a better poster because it kind of covers everything that we're doing. So you introduce yourself. Hi, my name's Lauren, and I tell you about our class project that we've been working on this semester for STEM Cats class. This is a group of 8 students shown here that are freshmen and getting involved in research. And our theme for the project was the effects of zinc on physiological processes, and we used invertebrate models, the Drosophila and crayfish preparations. Well, zinc is really important because it's an essential metal in animals, and in us, we need it to survive. It's a cofactor for many enzymes that allow these enzymes to function properly, so it's very important for us. But sometimes people might be taking too much zinc because it's in supplements that people can get from a health food store, as well as possibly exposure in the environment, such as people that are mining zinc, for example, or maybe work around galvanized processes, to galvanize steel. And then we thought maybe that this would be interesting too, because you think in the environment that maybe animals in their environment, maybe around zinc mines, for example, or wherever zinc processing is occurring, that they might be exposed to higher levels of zinc, and it might actually affect the animals in the environment. So we decided to test some of these properties on invertebrate models. Conducting some of our research, we found out there are problems, toxicity in humans with high levels of zinc. It accumulates in the brain, and it's correlated now with Alzheimer's disorders. It can affect cardiovascular function as well, and it actually affects bone development because it displaces manganese that is in the bone, and it can actually be incorporated in bone, and it might alter your calcium, the way the calcium forms bone itself. So we decided to look at a few preparations to see what it might do at different concentrations in Drosophila and in crayfish. In Drosophila, it's really nice because they develop very quickly, and we can look at the effects on development. So if they lay eggs, for example, in just one day they turn to a first instar, and another day a second instar, and a third day another instar, and then 2 days here they will actually turn into pupa, and then the pupa turn out to be adults. And we can tell these different stages by looking at the mouth hook formation, and that helps us in case they didn't develop well or they died. We could actually look at what stage they died in. We're also interested in doing some behaviors, how well they might respond to different behaviors. So you can have them crawling, and then you touch them, say on the abdomen, in the middle with a little bristle, and what kind of behavior do they exist. So compared to controls, do these animals fed, say, zinc at different concentrations, would they show different behavioral paradigms? That's one thing we're looking at. So we started with different concentrations of zinc in the diet that we fed them. So we took first instars, these little first instars, and put them into vials. We put 10 of these first instars into each vial, and we had 10 vials of each concentration. So we have 100 larvae in each one of these. So each one is actually 10 larvae to start with. And we see that all our controls were going pretty well at this concentration, and then at different stages they turned into pupa. So these different lines are the 10 different vials, the time, how many pupa formed over the time, and then of those pupa in each one of those vials, did adults emerge? And we see that we have basically 10 pupa in every one of them, and we have 10 adults in every one of the vials, but there was some scatter in the staging. When we feed them a low concentration, it actually seemed like it might have sped up development a little bit, because we're going back to look to see if this is a glitch or not, but it looks like they developed a little bit faster and more uniform, as well as the pupa all emerged pretty much at the same time. But when we start to go to higher concentrations like 5 millimolar and 10 millimolar, we see a delay for them to make pupa, and we actually see that there's fewer pupa at the end. A lot of them died either in first or second stages before they made pupa, and then of the adults we see fewer adults coming out of the pupa cases total. So It's having an effect at higher concentrations fed in the food. We still want to try this 20 millimolar, very high concentration, and see if it maybe kills more of the first instars, and maybe they won't even make pupa. So this is work in progress. We also fed adults zinc, so we just put 10 adults in 10 vials, in 10 different vials, so we had 100 for each one of these concentrations. And these are the survival curves over days of actually the adults. And our colleague at Benedictine University is doing this statistics on this for us. And so we can see at real high doses, they die off quite quickly, the adults. And at lower doses, real low dose, we didn't really see that much effect. But at 5 millimolar, there seems, at 10 millimolar, sorry, the blue line, that we're starting to see a big shift from the lower concentration. And we're looking at controls too. We need to put controls in here. We probably think it's going to be around here. To understand why some of these Drosophila larvae might die, we actually open them up and look at the heart directly. The heart is myogenic. It beats on its own. It depends on calcium coming into the heart for it to beat. So we open up the Drosophila larvae and we count the heart rate by counting the beating. And we put the different concentrations of zinc on, and this is what we found up here. We find out that when we put zinc on it, it depresses the heart, and the higher concentrations, it depresses it really fast. But we can get it back. It's not totally dead. In some of the preps, we can wash the zinc out, but it's kind of hard at the higher concentrations to get it to recover. Lower concentration, it seemed to recover a lot better. Then we looked at those behaviors that we were looking, not just these behaviors, but some other behaviors. And the two general behaviors that's used in the Drosophila world are body wall movements, how much they crawl. So we can count their body wall crawling every, say, minute, and at the different doses of zinc, and this is what we found at the different concentrations. More scatter over here, some of them are lower over here. We need to increase our sample size. Another standard behavior is a mouth hook assay. They have these two little mouth hooks in the front, and they keep eating. So it's a different neural circuit that does that. And we're looking at the higher concentrations. We need to increase our sample size, but we're beginning to maybe see a little trend. But this is only feeding them for 24 hours of the zinc and then doing the behavioral assay. We switched over to crayfish too, because crayfish are really interesting because they're like centils of water quality, and a lot of people use crayfish for different experiments of toxicity in the environment. We wanted to see what happened with them directly. When we inject zinc into the animal, we actually found out, up here I didn't show the data, but when we inject zinc into the crayfish hemolymph at different concentrations, at 10 millimolar, they would just stop. Their heart rate would stop, and they would die relatively quickly. They were kind of paralyzed from it, and then they didn't come back. And so we were wondering, how did that work in the crayfish? Well, we decided we could actually record from sensory nerves in the crayfish. So this muscle receptor organ, it has sensory endings that are embedded in a muscle, much like our muscles with muscle spindles that detect our muscle stretch. And we can move that joint right here, and we can record the neural activity with the suction electrode. And what we found is that the neural activity completely shuts out with 10 millimolar zinc. And at 5 millimolar, it also reduced the activity, but it took a little bit longer for it to happen. So how is zinc inhibiting sensory nerves? Is it the sensory endings itself that detect the stretching of the muscle? So these are stretch activated ion channels. Or is it altering the conduction down the nerve? That's still something to be addressed. We then went over to the neuromuscular junctions of both preparations, the crayfish and the Drosophila larvae, because they're very sensitive to different calcium in the media, because the presynaptic nerve terminal requires calcium to come in for vesicle fusion, for synaptic transmission. So the two preparations that we used here, one's the crayfish opener muscle that opens the claw, and we're looking at this amplitude of the 25th pulse for quantification. But here's some of the preliminary data. We have saline, we stimulate it, and it facilitates. It gets larger and larger. That's the synaptic response. These are stimulus artifacts from the stimulation, but you see the depolarization of the muscle fiber. We put the zinc on at 1 millimolar, it shuts down really fast. And you can see it's almost gone to nothing. And then we wash it out, it comes back. So that was pretty good. That's the crayfish. The Drosophila preparation is a little bit more difficult in the dissection, but we open up the Drosophila and we cut the nerve, put that into a suction electrode, stimulate it while we're recording a muscle fiber. And this is what we found here. We can see these evoked responses, like shown here. And this preparation is very high output, so it makes a big synaptic response. These are not action potentials in the muscle of the crayfish or the Drosophila larvae. They're graded responses. So what happens here is we can see a nice response. We put the zinc on at low concentration, it dampens it really quick, and then we wash it out, it begins to come back. These little changes in the baseline right there, that's where we wash it or change the bath. That's why that's shown there. And then if we go to a higher concentration in Drosophila, we see, yeah, the muscle depolarizes a little bit, but the amplitude of the event completely goes away, and it slowly comes back with washout. So that was really interesting. So in general, we think that zinc, is having a lot of different effects, and how it might kill an animal or alter their physiology is probably contributing to lots of things. But we know it slows down the development of Drosophila larvae at the higher concentrations. It can depress the cardiac function in both Drosophila larvae and in the crayfish, and it can dampen synaptic transmission, and it's dose dependent. Maybe for the sensory neurons that are over here, maybe it's blocking the stretch activated ion channels that allow the sensory ending to detect the stretch on their endings. But it might also be altering the neuronal activity, like the action potential traveling down the nerves. So that's a possibility as well. We hope to continue some more of this research and actually follow up on some of these details, and then increase our sample size so we can get some idea of maybe how it's working. So it would be nice to know if zinc is primarily targeting the calcium channels or not. And maybe a way around that is to raise the calcium and then give different levels of zinc and see if the higher calcium displaces the function of that zinc, like if they're competing for the ion channel or not. It would be nice if we could record from single channels and know more about these particular calcium channels in the nerve terminal and on the heart, and if zinc is blocking the channel itself. But that would be more difficult of an experiment to do, and maybe by getting this information out there, somebody that has patch clamping techniques could actually answer those questions for us. Thank you.